Hi, Agarose gel electrophoresis is a procedure used to separate DNA fragments based on their molecular weight, that is in turn because of their sizes. It is an intrinsic part of almost all experiments in molecular biology and biotechnology. There are three steps involved in this practical. Number one is preparation of agarose gel. Number two is electrophoresis of the DNA. Number three is visualization of DNA fragments. This is the setup of gel electrophoresis. There are two units in this. This is the gel tank with agarose gel. This is the power supply. These two are connected by these two cables, and the gel tank has two electrodes, one this side and one this side. This is positively charged cathode, which is indicated by red color. This is negatively charged anode, which is indicated by black color. So there are two cables. One is the black cable, which is anode, and the other one is a red cable, which is cathode. So you have to connect in this fashion only when you start electrophoresis. When the electrophoresis begins, when you switch on the power supply, the DNA moves from cathode, which is negatively charged, towards anode, which is positively charged. Because DNA is actually negatively charged, it will move towards the end, which is positively charged. So you can see the DNA fragments which have moved, and they are moved based on their size, and they move towards the positively charged anode. The most crucial thing in electrophoresis is loading DNA. You have to load DNA into the well carefully without smudging. You can have an equipment where you can load multiple sets of DNA samples simultaneously, or you can use micropipette and load each DNA sample into the well specifically, carefully, without smudging. The gel in which the DNA samples have run is placed under transilluminator, UV transilluminator, and you can see the fluorescent DNA fragments in the form of bands. So these are the bands that appear under transilluminator as fluorescent bands. Purified agarose is a powder which is insoluble in water or buffer at room temperature but dissolves on boiling. In fact, agarose is a linear polymer extracted from seaweeds. Electrophoresis is a technique used to separate charged molecules in a gel. DNA is negatively charged at neutral pH. When an electric field is applied across the gel, DNA migrates towards the anode. Migration of DNA to the gel depends on molecular size of the DNA, agarose concentration, conformation of DNA, and applied current. Visualization of DNA fragments is usually done under UV light. Under UV light, DNA appears fluorescent. The process of gel electrophoresis is monitored by observing the migration of visible dye. Two commonly used dyes are xylene cyanol and bromophenol blue, which migrate at the same speed as that of the DNA fragments based on the size, 500 base pairs and 300 base pairs respectively. These tracking dyes are negatively charged. Low molecular weight compounds that are loaded along with the sample at the start of electrophoresis. When the tracking dye reaches the anode, electrophoresis is terminated and DNA fragments, the gel as such, is kept under transilluminator, and the fragments are visualized under UV light. Materials are required for gel electrophoresis are as follows: agarose powder, TAE buffer, then Ethidium bromide, DNA ladder, horizontal electrophoresis apparatus, power supply, transilluminator, controlled DNA, and DNA samples. The protocol for gel electrophoresis. In the first step, we are preparing 1% agarose gel. Prepare 1x TAE by diluting the appropriate amount of 50x TAE buffer. we approximately require 200 ml 1x TAE, dilute 4 ml of 50x TAE into 20 ml distilled water. Weigh 0.5 grams of agarose and add 50 ml 1x TAE. This gives 1% agarose. Boil till agarose dissolves completely and a clear solution is obtained. Meanwhile, place the comb of electrophoresis set in such a way that approximately 2 centimeter it is away from the cathode. 2 microliter Ethidium bromide to the molten agarose. Pour the agarose solution into the central part of the tank. When the temperature reaches approximately 60 degrees, do not generate air bubbles. The thickness of the gel should be around 0.5 to 0.9 centimeter. Keep the gel undisturbed at room temperature for agarose to solidify. The next step, that is electrophoresis. Pour 1x TAE buffer into the gel tank till the buffer level stands at 0.5 to 0.8 centimeter above the gel surface. Gently lift the comb, ensuring that welds remain intact. Connect the power cord to the electrophoretic power supply according to the convention. The general practice is red is anode, black is cathode. Load the DNA samples in the well in the desired order carefully, and this is very important. This loading should be done accurately, properly, without smudging. Set the voltage to 50 volt and switch on the power supply and allow the dye to run on the gel and observe the dye that is moving on the gel. Switch off the power of the electrophoresis unit once the process of electrophoresis is complete. That is, bromophenol blue reaches till the end of the gel. It's almost 1/4 of the gel. This will take about 1 hour's time, and after electrophoresis, DNA samples can be visualized under UV light. They appear fluorescent. The smaller fragments would have traveled the longer distance, and the larger fragments would have traveled the lesser distance. By this method, by this technique, we can separate DNA fragments based on their sizes. There are 2 precautions that you must consider. Number 1 is Ethidium bromide must be handled carefully. You must wear gloves when you are using Ethidium bromide, because it's a mutagen and a carcinogen. And the next important precaution is ensure that the gel is oriented in such a way that the welds are placed near the cathode and away from the anode. As DNA moves towards anode, the red terminal is anode and the black terminal is cathode.
Thank you for watching my videos. Please like, share and subscribe. We'll meet again in the next video with some other topic. Thank you very much.